Hello and welcome to this International NASH Day panel on NASH Diagnostics and beyond. I am Donna Cryer, the CEO and founder of the Global Liver Institute, the largest patient advocacy organization in the world for liver health. We bring together more than 200 organizations across our councils, campaigns, and events like today on International NASH Day to advance liver health as public health and to both prevent liver diseases and scale optimal approaches to improving the lives of individuals and families and communities impacted by liver disease. Today's panel, I am excited to bring you a world-class team of experts to discuss this issue of NASH diagnostics and beyond. I have with me Professor Quentin Anstee. I have Dr. David Diehl and Dr. Mazen Noureddin, whose extensive biographies I could take up our entire time reading, but I would rather turn to the conversation. So, gentlemen, welcome.
Thank you.
Thank you for having us.
So the first question I'll address to you, Dr. Noureddin. We have seen recently, Diehl has worked with AASLD to have the American Board of Internal Medicine go through their procedures and announced in May that the requirement to achieve competency in performing liver biopsies will transition from an opportunity to train for their transplant hepatology fellows to an option. This preserves the ability for fellows to learn biopsy if desired, but aligns with the feedback and the changes that we've all seen to align practice more towards non-invasive diagnostics. So does this major milestone in training indicate that we've gotten beyond the biopsy?
Well, first of all, Donna, thank you for having me here. And it's an honor to be here on NASH International Day, and especially with GLI that has done significant work in the field of liver disease and awareness, and in particular NASH, that is close to my heart. Thank you for your work with the AASLD and the Internal Medicine Society on this. Although hepatologists still like liver biopsy, and it gives us a lot of information, but there is a realization that with now current liver diseases that they're so prevalent, such as non-alcoholic fatty liver disease, affecting the 25% of the population, it has become clear that if we just go do biopsy all these people, we will have complications, and many of them. And that led to the world of non-invasive testing that Quentin and I work on extensively and look for the important features of liver disease, such as fibrosis, that now we can easily detect with non-invasive testing. Thus, I think it is a step toward the right direction that they are still giving them the option to learn this important method, yet open their mind that there are many other tools that they can use and that have been around and that have been validated and showed us accuracy in disease severity, disease progression, and correlations with outcome. So
that was a bell announcing a new milestone in us getting beyond the biopsy. It's happening so fast.
I'm so glad that this step has been taken, and hopefully we'll see less biopsies and see them only done when it's needed.
Absolutely. Dr. Anstee, you lead probably the largest global consortium around validating non-invasive diagnostics and helping to evolve the field. And so tell us, where do we stand now? How close are we to really, truly getting beyond the biopsy?
Yeah, well, again, Donna, thank you very much for the invitation to participate in this. It's a really exciting day to raise awareness of fatty liver disease. There are a number of global efforts to really get beyond biopsy. So you're absolutely right. In Europe, there's the Litmus Consortium, which I coordinate. In North America, there's our sister organization, Nimble, and both of these are working very closely to try to generate the evidence base we need to robustly move away from biopsy. I think we're making great progress. We've had letters of intent accepted by both organizations with the FDA, which is an important step towards biomarker qualification in clinical trials. We're also generating robust data showing how these biomarkers actually perform. And that's one of the key things here. Historically, somebody's invented a biomarker, they've been very enthusiastic about it, they've published some data to show it works very well, and everybody feels very good about it. But what we need to do now, and what we are doing in Litmus and also in other efforts like Nimble, is take the biomarker, independently apply it to a very large cohort so you can truly understand how the biomarkers perform and then use that to tune decision making. And that's a key step as these technologies mature.
Fantastic. Dr. Diehl, we don't want to get too far ahead of ourselves. Biopsies are still necessary in some circumstances, and there are efforts to improve them, smaller needles, all various things. And so help our listeners understand when biopsies would still be used and why, either in a research setting or clinical setting.
Sure. Donna, thank you for inviting me to this discussion. So I agree with the other panelists that non-invasive testing is clearly necessary to address, the huge number of patients who need an accurate diagnosis. However, biopsies, let's face it, biopsies are still going to be needed in a group of people. I don't see them going away. And certainly we'll be talking about maybe new agents that are in the pipeline for treating NASH. I imagine in many situations, a biopsy will be required prior to starting someone on a lengthy course of treatment with a new pharmaceutical, for example. You're right. We talked about how the skill of liver biopsy has, the reality is it's being done by interventional radiology largely now. There may be a few senior hepatologists who still enjoy. I don't know if anyone enjoys doing the liver biopsy.
I know, big poke in the side. They look at me and they're like, "Oh, we see. We don't even need to. We see where people have been in before. There's some market X, and we go right in." So.
Right, but so we've, where I'm at, we've largely switched liver biopsy over to a endoscopic ultrasound guided approach, which, first of all, the yields of the tissue are great. There's a lot of data showing that they're equivalent to or even better than percutaneous. So as far as tissue yields, I don't think any hepatologist or pathologist is going to have an issue with that. Another thing is we can get bilobar biopsy, right and left lobes, which for NAFLD type diseases may be advantageous. There'll just be less sampling error. And another advantage is some patients who need a liver biopsy also need an endoscopy for various reasons, maybe to look into their reflux, they have some dyspepsia, some stomach symptoms, or in people with significant liver disease, we may want to make sure there's no esophageal varices. So people who need a liver biopsy and an endoscopy, they're very well served by an endoscopic ultrasound biopsy. And the other major advantage I want to point out is that it's really the most humane way of doing a liver biopsy. Patients are sedated. It's a very quick procedure, very safe. Safety profile, very, very similar, I would say, to percutaneous liver biopsy, if not better. I have had some patients who have had a liver biopsy done percutaneously, and then we did it by EUS guidance, and they were very pleased with how that went.
Thank you. That's Quentin. Go ahead.
So I think this is a really interesting point. And one of the things David said there, it chimes, which is quite important. This isn't a case that biopsy will be removed from the menu of tests. When you're looking at diagnostic uncertainty and wanting to clarify what a diagnosis is, a non-invasive is not necessarily your way forward for that. What non-invasive testing does is it helps to accurately grade the disease. So in other words, differentiate steatosis from steatohepatitis. But even better, it helps to stage the disease and determine the amount of fibrosis. But if you're asking the question, for example, is this autoimmune hepatitis or liver disease, that's where a biopsy comes into play. Now, so there is still a place for it. The other point, and a counterpoint to what David's saying there, though, is that biopsy is not scalable. We're dealing with a condition that affects 25% of the adult population in most of the territories who'll be listening to this video cast. That is a huge number. And there is no healthcare system in the world that could actually biopsy that number of individuals, even if we wanted to, and clearly we don't. So we need to think about this smartly. There's a place for biopsy, and we can discuss and debate what the right route to do a biopsy is in that situation. But more importantly, for the majority of our patients, what we need to do is accurately grade and stage them based on their clinical probability of disease, and that is something we can do non-invasively.
Thank you. And Mazen, I was going to ask you, so what should a patient expect in most cases? I've seen, the elaborate flowcharts that now we're providing to primary care and others as this gets to Quentin's point, well beyond just hepatology, to be able to reach all of the patients who we suspect or at risk for being somewhere on the NAFLD or NASH spectrum. So from a primary care perspective or a patient perspective, what are they most likely to experience? Blood work, imaging, in what sequence?
Yeah, so I think we have made quite a bit of advances in society guidance in terms of this area. So what we will likely see in the next few years, more patients to be screened. And those are the high-risk population, as was mentioned by two documents recently published by the EASL, European Association for Study of Liver Disease, and the American Gastroenterology Association. So EASL has recognized the need for screening high-risk population a few years ago, and AGA just added that recently. And the expectation that patients with type 2 diabetes, especially above a certain age, like 40 or 50, now will be benefiting from screening, meaning a patient who comes with type 2 diabetes, his primary care should now tell him, hey, we're going to look for if you have fatty liver disease or not. And the way that the patient will have this, by just having his blood routine tests, which is CMP and CBC that the patients don't need to know about, the doctor should calculate a score, which is called the FIB-4. And that FIB-4 will determine if the patient will stay with the primary care physician or go to hepatologist or need another test. And that another test will be likely to be another test, which is imaging test, such as transient elastography, or be a blood test. And there are many that have been talked about, for instance, the ELF test. So the patient's expectation that their health will improve by looking for a silent liver disease if they have the risk factor, and they will start hearing about a score looking for fatty liver disease or not, and they might need additional non-invasive testing. And like down the road, they might need MRI or even rarely liver biopsy, as Quentin mentioned. But with a couple of simple non-invasive tests that they're quick and easy to do, now their health will benefit, and we hopefully will not see any more that patients present to us with full-blown cirrhosis.
And that really is the key, to sort of pull back the lens and make this more of a public health issue, a prevention issue, an early stage issue, so that it doesn't, so many people don't present for the first time in the cancer center or needing a transplant. That's when we know these efforts will be successful. and thank you for listing off so many of the different types of tests that have been evolving. I think from when I talked to primary care, that was their initial reluctance about engaging with us in NASH, because they were saying, is there an HbA1c? Is there something simple? Is there one test? To help us fit this into the many things that we have to do in primary care. And we're getting closer to that, or at least getting closer to saying, in under what circumstances you should be able to use which test.
And Donna, my example is, I think the FIB4 is easy and straightforward to do. And if you're looking for retinopathy through an eye exam, and if you know how to do it, if calculating FIB4 is much easier in a diabetic patient.
Absolutely. I think one of the key things here is that it really is, as Mazen said, a multi-stage process. The first step is to recognize the risk. Obesity, diabetes, high blood pressure, high cholesterol. The more of those an individual has, the greater the chance they've got significant fatty liver disease. But then to come in with a test like the FIB4 that has what's called a high negative predictive value. So you can immediately apply the FIB4 test and say that 70, 80% of the people who might on the face of it be at risk do not currently have significant liver disease. That's a really crucial point, because you can immediately reassure those individuals that they have it and save them further anxiety, save them further investigation. And then you home in on the remainder using a staged approach, as Mazen was talking about, potentially using something like FibroScan, which is an ultrasound-based technique, or MR elastography, which is something that Mazen's really been working and evolving, and a range of other potential biomarkers to further focus down on the individuals most likely to have advanced liver disease. And all of this can be done in a way that is highly acceptable to patients, because it is quick, painless, and safe.
Absolutely. I actually had pointed out that looking at some studies, I'm like, it looks like a tape measure is probably our most scalable non-invasive diagnostic to take waist circumference first. So everybody dig that out of the drawer and take your waist circumference first, and then but also in terms of ease, I having access to my own lab work, I'm able to calculate a FIB4 right from publicly available calculators. So in terms of being able to bring this to a place where we can use what we have, and to be able to really start the assessment for people in those high risk categories that you so articulated, we really are there. And as we talk about, Stop NASH Now and working with things that we can do today, even someone who just has access to the lab work that they've already done can calculate a FIB4, as well as it being integrated into many hospital EHRs. Dr. Diehl, let's bring you into this conversation. What does this look like today, and then what is the next step you'd like to see?
Well, I'm interested in hearing if there are new blood-based biomarker tests that are coming down the pike, which would help figure out, narrow the number of people who might end up needing a biopsy. The current guidelines, it's certainly, if someone is on one end of the spectrum, like low probability of having NASH, for example, they're safely able to be managed without a biopsy. And perhaps someone on the far other end where it looks cirrhotic, again, we could avoid a biopsy. But there's a large middle group that needs another level of differentiation. And so I'm wondering if there are some blood-based biomarker tests that look promising to you.
And there are, and I'm going to borrow from the recent guidance documents just to stay evidence-based. So you're right, sometimes people might not, that middle range people, and what we're talking about, this FIB4 sometimes cannot really determine if the patient has it or not, and this is about 30% of the population. There are blood tests that have come out. So in the AGA guidance, for instance, The ELF test was not available when the guidance came out, though they mentioned in the document that when ELF becomes available, it's an alternative to a FibroScan. Again, as Quentin mentioned, you start with FIB4, and it has a great ability to say yes or no, this patient is in trouble. And those that they fall within what we call it the gray zone, that we really don't know, or we still don't know, they can get another blood biomarker test. And there are some available, like the one I mentioned, and there are many in the pipeline. We're trying not to get, study like 20,000 of them, but they're really 2 to 3 promising ones that the work by Litmus and Nimble, they're teasing them out, and they're related to the pathophysiology of the disease, and we might see them in the near future, and that will improve the diagnostic performance of what we have now. So we're there, we're doing really good, but we want to excel and do better, and
So
that's the stage we're in.
I very much agree with you there. I think the big thing to remember here is that what's the phrase? You don't want perfection to be the enemy of good. We have good non-invasive tests. It's about applying them. We can do a job fairly successfully with what we have already easily available to us. For sure, consortia like Litmus are going to improve on that and innovate. That's one of the things we're there for. But there are already tools available, and we just need to apply those in practice. And this is something that the GLI under Donna's leadership and the work of people like Jeff Lazarus and so on have all come together to really help us drive forward that public health agenda to get these things moved into practice, not just in Mazen's clinic and my clinic, the diabetes clinics, in the primary care setting, all the other places where we know that individuals at risk are, but are as yet undiagnosed with advanced fibrosis.
So, thank you for that. First of all, so that's in a practice setting where I absolutely see the momentum and movement. With the various initiatives, whether Nimble, of which we've been proud to be a member, Litmus, of which we've worked very closely, and now the nail knit study, Mazen, that we lead. How close are we to being able to have clinical research without liver biopsies or with minimal liver biopsies? How close are regulators to accepting that? Because certainly that will expand the number of people who would want to participate in a NASH trial.
That's a great question, Donna. And as I said earlier, and the Litmus and Nimble have done outstanding amount of work to get us closer. I think we're much, much closer than 3 to 4 years ago. The regulators, rightfully so, they just want to make sure when they replace liver biopsy with non-invasive testing that they have confidence that they are giving the right decision if pharmacological therapies are working or not. And this has been done in two folds. Quentin has done great work on showing the downfalls of the biopsies and why sometimes that this reference standard that was great a few years ago and we didn't have another option, now it is less feasible and has its own problem, while NITs they are excelling in a way, especially if you combine them. So I think we are close. We just have to show the last part through these consortium and other ones that they are supporting them and coming out to correlate with outcomes. And this is the regulators ask. If you want to ask me, I could be wrong. I wasn't interested in Quentin's opinion, but I think we are about 3 to 4 years ago, I hope. If shorter, that's music to my ear. If longer, I will not be surprised. But this is the time frame I will put for phase 3 trial to get rid of liver biopsy, hopefully.
Quentin, what do you think? What is your prediction or prognostication on how close we are?
Yeah, so I think we're making good progress, and I think Mazen summarized it very well there. We're already seeing that early phase studies, the 2A studies, they're a non-invasive trial now. it's biochemistry, it's circulating biomarkers, it's MR testing. So we've already started it there with the 2As. I'm increasingly seeing a move towards it in 2B studies. The pivotal phase 3 studies, it's a little way off. And just as Mazen, it's about producing that compelling data set, because the regulators have clearly signaled they're open to this change. But quite rightly, they need to see the evidence to back it up. We've demonstrated some of the limitations with histological interpretation, high inter-observer variation, the fact that the concept of ballooning and complete NASH resolution is no longer a scientifically sustainable study endpoint. And I think there's very few people in the scientific community who would argue against that at this point. What we've also got to do is provide the evidence to back up valid alternatives. The challenge is that the standard of evidence that was required to establish histology as the trial endpoint is in many ways lower than the standard of evidence we're now being required to produce to make change. So there's a bit of complexity to it there, but I think the great thing is there's a huge amount of goodwill from the point of view of the advocacy, from the patient perspective, from the point of view of the academic community, and crucially from the point of view of the regulators. So I'm optimistic about this within the next couple of years. And those letters of intent that Litmus will have had accepted by the FDA and Litmus' successful interactions with the European Medicines Agency really are the crucial first step in that process.
If I just might for a moment, having a committed regulatory partner who will agree to the endpoints and the standards and then stick to them is a really important part of this process, as this community is working so hard to develop this evidence. We need to make sure that when we hit the mark doesn't then move.
Absolutely, and I've got a lot of confidence that our partners in the regulatory agencies are, listening. They're responsive, and they are very perceptive in terms of what it is they need to see. So I'm very optimistic on that basis.
Excellent, David. I wanted to bring you in.
Yeah, I'm very intrigued about some research that I've been seeing on artificial intelligence to analyze biopsies. The current grade score, F0 to F4, whether using the Medivir scale or Ishak, it's a very coarse determination, and it's hard. It depends a lot on the pathologist's interpretation, and some of this artificial intelligence work that's being done quantitates the scar tissue, the collagen, the fibrosis in a much more granular way. So it may be easier to see an impact of a drug, for example, using a yet to be developed scale. So I'm very excited. I can't wait for these to come. But my question to you guys is, do you see AI assistance with liver biopsies on the horizon?
I'm going to weigh into that a little bit just to give some context, because I think it's a really important part of this discussion. As Quentin was talking about the integrator variability, I just want to pull back a bit for the patients and primary care folks who are listening. There aren't thousands of pathologists, being produced for the number of trials that are being done, the number of patients that are being screened, and even if there were, the differences in looking at a particular sample of tissue that we get from a biopsy can differ from the human eye, from the human interpretation. I know there are improvements in training pathologists to read better, to read biopsy samples better, but there are limitations to that. And so that's where AI comes in, artificial intelligence, to improve the quality of that process, to assist the pathologist, to assist physicians. And so when we talk about that category of we're still going to need biopsies in some situations, how can we make them better? How can we get more from the results? And how can we scale that part of it? I think AI is an important part. And so then Mazen and Quentin, how widely is that used? And where do you see that going?
So there are a number of different AI technologies, and we published on a few of these now. So The majority of them are still very much research tools.
Mhm.
They're used in the trial space, and they're used in the discovery science space. But they're moving, and there are active discussions, again, with the regulators
Mhm.
about moving to this. And it goes back to something David said, which I think is really important. The human eye is fantastic at diagnosing diseases. Is this NAFLD? Is this autoimmune hepatitis? But what we're not so good at is quantifying things. When we look at a tree, we don't say there are 3,023 leaves. We say there's a tree. So why are we asking humans to try to quantify when it's not what our brains are wired to do? And that's where the machines come in.
Mhm.
So you can do this either with a stain-free approach, and there are some of these like second harmonic generation microscopy, which is very useful because it's not limited to what standard histological stains can measure. So it gives us potentially greater granularity. And then there are ways of robustly measuring change or degrees using digital images of standard biopsies. And both of those techniques probably offer us something different. One of them is more akin to what the human eye sees, but with a computer doing the measuring. The other one goes beyond what the human eye can see and looks right down at the fibrils of collagen and the cross-linking. So potentially gives us even more biological relevance. I think we see these being used more and more in drug development, in terms of trials, increasing the robustness of endpoints as we move to non-invasives. And that's undoubtedly the direction of travel here. But it's something that's going to assist us in that process.
Fantastic. David, you also made some really important points about both the diagnostics and care for patients with advanced liver disease, with chronic liver disease increasing, and patients having needing upper endoscopy, needing lower endoscopy, having ascites, having different types of varices, having complications. I don't want to spend so much time on the early stages in the prevention that we forget about increasing number of patients in this time in the next few years who will be presenting with very advanced liver disease and with complications. And so, what does that look like in terms of diagnosing and caring for those patients with advanced liver disease and NASH?
Right. So, the thrust of our discussion today is to try to avoid patients getting that far. But it's still, we certainly in 2022, we see someone show up with a upper GI bleed related to esophageal varix, which maybe it's a patient who has never had a drop of alcohol and has all the stigmata of having a lifetime of fatty liver disease. So when that happens, it's rather an emergency situation. We have endoscopic means of stemming the bleeding, but certainly we want to get, figure out who is going to develop that and intercede before they get close to that.
And the fact that fatty liver disease, NASH, is a progressive disease is a very important part of the messaging, part of the understanding that we need to convey. And I saw in some of the presentations at Digestive Disease Week that progressors have worse outcomes. How are we doing at understanding who's going to be a fast or slow progressor? As we talk about, in some circumstances, you hear, "Oh, well, NASH is a disease that takes 20 years to manifest," and then you see other patients who go very quickly, from low fibrosis scores to high fibrosis scores and complications. So how are we doing on understanding who are the progressors and what happens to them?
Donna,
Quentin, okay, Mazen, yes.
no, actually, Quentin has done a lot of work on this. I want to comment on it from PCP kind of
Okay.
point of view, and there's a lot of work being done on the genetic side of non-alcoholic fatty liver disease, and Quentin has excelled in this work in particular. So what we can tell the primary care, there are few SNPs or genes that are related to disease prevalence. severity and possibly progression. For instance, I will name one of them. It's called PNLP3. That it's a little bit early to put it in the clinic or ask primary care to use it, but eventually we might see people subclassified based on these as rapid or slow progressor, and that would not make it more complicated for them. I mean, with hepatitis C, we had genotypes. I don't see why not. We can identify patients that will progress faster or have more complications based on genetic testing. I will keep it as simple as this for now because the field is booming and there are so many of them. There are good genes, there are bad genes, and indeed, the good news, they are. Quentin and I were in a meeting this week. There are therapies that they target the genetic components of the disease, which is very exciting in a precision medicine and personalized medicine approach.
Right. I would be very remiss, being the wife of a geneticist, to not mention genetic diagnostics as part of this. So, Quentin, please continue this part of the conversation.
No, absolutely. I mean, I think Mazen's done a fab job of summarising this. Yes, we already know of certain genetic modifiers that increase the risk of having more severe disease. Now, the thing to remember about them is that fatty liver disease is what's called a complex disease trait, which basically means it's not one gene causing it. That's like cystic fibrosis. You have the disease. Here, we're probably talking about 10 or 20 genes, all acting in combination, some of them pushing it to be worse, some of them making it a bit milder, and then also influencing how we respond to our lifestyle choices. What did we have for lunch today? Those sort of dietary factors and so on all get thrown into the mix. What that means is that the effect of any one single gene is modest. It may increase your risk a couple of fold, maybe fold at most. That's still important. It's still worth targeting. We're not quite there with rolling those out into the clinic at this point. Huge amount of active research going on the genetic factors. And there's one other area that I think it's worth thinking. This is really exciting. Right now, because we're based with histology, we talk about liver disease as if it's one disease. As we start to look at gene expression levels, what's called transcriptomics within the liver tissue of a large cohort of patients, and this is something we've published on, it's also been published by Arun Sanyal in a separate cohort of individuals, you begin to see there are subsets of patients which aren't apparent on the biopsy. When we look at the genes, we can see that this little group over here is different to this over here. Now, that's telling us something about potentially what the processes are that are driving the disease. We need to understand that better to be sure. We also need to understand what that means in terms of long-term outcomes. But we're getting the clue that it probably isn't a one-size-fits-all in terms of how we diagnose, how we risk stratify, and how we treat. And that's a very exciting first step towards personalized medicine.
There's an unusual group, sometimes referred to as non-obese, fatty liver disease. Are those cases, do you think, have a genetic basis compared to the metabolic basis to the others?
Yeah, so we've published a couple of cohort studies on that in the European NAFLD registry. It's very interesting that the non-obese individuals appear to have very similar disease natural history compared with the individuals who are maybe carrying a little bit more weight. So that's the first thing to say. As yet, we don't have strong evidence for a particular genetic variant as a driver for it. But what we do know is that certain genes appear to act more strongly the more weight you're carrying. So it actually works the other way.
Well, another aspect of it is that we see when we have cohorts of patients out of Singapore, for example, or other Asian countries, that they're presenting with fatty liver disease and NASH at lower weights, lower body mass indices, and so I think that speaks to, one, the importance of diversifying the clinical research participants so that we really understand, as you say, the many types and perhaps profiles and, of diseases that may come under this banner of fatty liver disease and NASH. But it also may point us to different different genes or different triggers for gene expression. And so I think that that's David. You ask a really important point about how we get to the answers.
And I want to also add India,
Yes.
which we're going to translate to. There's a huge lean NAFLD and NASH in that population and their data from that country. So I cannot agree more with you, Donna. We have to include minority in the disease. For instance, there's data showing that Hispanics participate less in clinical trials. Indeed, it's on the low side why they have a high prevalence of the disease. We have to include countries, such as Asian countries and India, in our clinical trials.
Yes, it's very state, county, country specific as to who's considered a minority. And so, in as much as NASH is a global disease, we need to make sure that we're looking at diversity in our clinical research in a global setting. And so, I know that is one of the reasons why our how to participate in a NASH trial or materials are now in 16 languages to try to reach out to patients and encourage them to talk to their doctors about this opportunity. What else can we do to help people understand the importance of participating in NASH clinical research and make it easier for them to do so? Quentin, how could we double the size of the number of people in your registry?
Yeah, absolutely. I think that's a great question. Wouldn't that be wonderful if we could? It would really help us to move the needle in the direction that we all want to do. I think the first thing to say is that taking part in clinical research, patients usually do better as a result of it, simply because they see their doctor more often, they're spending more time in that care environment, and then it's not just about the disease that they're in the study for. It's also about the fact that the colorectal cancer is picked up earlier, or the mild anemia that otherwise may not have been even noticed is picked up long before people become symptomatic with it. So there's an altruistic aspect to it. Please help us; it'll help the world. The second aspect of it is there may be something for the individual as well, because they are being looked at and they're seeing people that bit more frequently.
Absolutely. I believe I remember sitting next to you at Paris NASH years ago, looking up at some of the results, and the placebo group was doing pretty well compared to the treatment group. And I was like, is that the effect of, care? Since so many liver patients aren't connected to care. And then with the opportunity of participating in a clinical research study, they're getting a lot of care and attention for the first time, and that's improving their health. So I think that is part of it. It does beg the question: how do we standardize the placebo group so that we truly can see what are the effect of the intervention rather than just of care? So are we standardizing are we standardizing standard care and making progress in that, either through the liver forum or other means, so that we can see the true effect of the interventions and trials?
There is some work, Donna. So the placebo group in particular is complex and unpredictable. Part of it could be genetic components, environmental components, but there have been multiple publications on the rate of progression and regression and meta-analyses on that from clinical trials. There are more efforts, as you said, through liver forums and others to collect data from many sponsors to study this in more depth. Also, there was a document on standardizing what to do with the placebo patient that came out from the Liver Forum. I think Dr. Abdul Malik is the senior author on that one, and discuss what can be done in minimum in terms of lead-in phase or no lead-in phase. There are different categories on that, and there are publications. There's more work to be done, but there's tons of work on the placebo. But I want to emphasize on the point you said very eloquently, that patients, even if they get just randomized to a placebo, we know more about them. We know their disease state. We know their fibrosis state, and many of them get better actually, which makes sense, right? You go to the doctor, you're on the scale, you start eating better, exercise more. So
what get measured gets changed. Dr. Diehl, what would make the most difference in research coming out to the way you practice and the patients you see?
Well, Quentin and Mazen are, nobody knows more about, the pipeline of agents that we might be able to see. But as someone, not privy to all that deep information, I always wondering how come it's taking so long to see new agents to treat this. And certainly, people need to address their metabolic issues. But just to say, oh, lose weight, it's too simplistic. It's hard to do. And where I'm at, we have a very robust nutrition service, as well as bariatric surgeons for those patients who are interested in pursuing that. So a question is, outside of clinical trials of these agents that these doctors are working on, what other than generic advice, lose weight, what can a patient do?
I think you brought up several examples, and then Mazen, I'll come to you. I think that is really important to emphasize, that between access to a nutritionist, a dietitian, weight loss medicines, bariatric surgery, there are gastric balloons and sleeves that have an actual indication for NASH. And so in that space between advice and surgery, there are actually procedures that do have data on NASH and that people can have access to right now. So I think that's a really important part to this conversation about what we can do today and what practitioners can recommend for their patients. And we're seeing that integrated fatty liver disease clinics are involving more bariatrics and procedural balloon interventions and things like that for their patients.
Regarding bariatric and endoscopic interventions, there are several out there, and one of the major rate limiting steps, at least in the United States, is they're generally not covered by insurers. And like where I'm working, patients don't have $15,000 or what it is to get a endoscopic gastroplasty, which might make a profound impact on their weight loss. So and I'm not recommending everyone have a major surgery for that, but that in between the endoscopic methods, I would love to see more support mainly from the insurers about those approaches.
It's definitely something that we're working on in our advocacy, and why we're excited to have the Obesity Action Coalition and the Obesity Medicine Societies involved in our NASH Council to make sure that patients do have coverage for all these elements that we have collectively as a field decided really should be options for them. Mazen, what are you seeing?
I want to bring optimism to this. And David, you said, how come we don't have a drug approved? I don't think NASH is different from certain diseases and the length it has taken to find maybe approved drug. It could be more than other diseases, but there are many diseases that it took them some time to approve. Example from our world is hepatitis C, and I think we'll probably get there in a sooner time than hepatitis C duration for therapies, hopefully. Now, what I want to say, there are many exciting drugs in the pipeline, and we might have drug approved in the next couple of years. It took some time, probably because of the reasons we talked about, like biopsies variabilities and mastering the field, finding AIs, moving eventually to NIT. So it's just going to get better and better. Nevertheless, there are exciting medications in the pipeline that affect the liver directly, such as FXRs and TH betas, and some of them might get approved in the next few years, but also chasing them very closely. Drugs that are already on the market for other indications, such as type 2 diabetes and obesity, and those are GLP-1s, for example. We have good data in the NASH field that they work well, and we're just waiting for the phase 3 trials. And also, for instance, and if you take the AGA most recent document, they encourage using, for instance, GLP-1s, not necessarily for the indication for NASH, but to manage comorbidities, which is very important in these patients, such as type 2 diabetes and obesity. So they are now, for those that they say, well, if I discover NASH, so what? One, you don't want to say that because you don't want to miss cirrhotic or HCC that is hiding in a patient. And two, you actually can make a difference by using, even before endoscopic procedures or surgery, medications that they can modify comorbidities such as type 2 diabetes, obesity, that even have downstream effect on the heart. So I really see the glass half full, and maybe started filling more than half.
I feel optimistic as well, and I'm very excited to see that the American Heart Association has weighed in with their scientific statement about the connections with cardiovascular disease and NAFLD, ACE, and their new guidelines, NCQA, and its new white paper. So I think we see a momentum. So, Quentin, the question I want to turn to you is there perhaps a silver lining in the delays that we've seen in drug development, that we've been able to have so much evolution of all these other parts of the diagnostics, of our understanding of the disease, so that when a drug is finally approved, hopefully in the next year to 18 months, maybe I'll hope be hopeful, that we are a better prepared field to do all the things around a patient that needs to be done when a drug is finally approved.
Yeah, so I think you've put that very well, because of course, if an agent is available, the key question is how do you find the people who are going to benefit from it? If you put it in the tap water, you diminish the benefits, increase the risks of side effects. So therefore, we need to target the agents to the right people. And you're right, we can't biopsy everybody. We don't want to biopsy everybody. We need the non-invasives to be mature enough to actually help guide us in that way. And there's been a huge amount of progress in that respect in a few years. So I think we're now in a much better place to say that actually we have the right tools to get agents, when they become available, to the people who are going to benefit from them. The step now and the challenge is to put those pathways in place. It's not enough to have the tool, but also you've got to implement it. And we've got to put those care pathways in place. All the people are being identified in the community and passed forward so that they're in the right clinics to benefit when these agents do become available in the next, hopefully, few years.
Absolutely. And David, to your point about making sure that there is a partnership with payers, and opportunities that we have now in this time to increase their understanding. We'll be having a payer summit next month, I believe, in the relative time for when this airs, to educate payers about NASH, and for patients to educate payers about NASH, and for payers to talk to us about what their needs. But in my conversations with them, it has been very much about how do we appropriately risk stratify and segment these different patient characteristics so that we could align the appropriate benefit packages and treatment options for them. And I really don't think we were ready a few years ago to answer their questions with the level of specificity that would have been satisfactory. And I think we're much closer today to having those satisfactory answers to payers. So as we round this out on NASH diagnostics and beyond, we've talked about a range of things. We've talked about genetic diagnostics. We've talked about serum-based diagnostics, imaging, and ways to make biopsy better. So what do you want to have, each of you, and I'll go with David, Mazen, and Quentin. So whether to primary care or to endocrinology or other practitioners who are just starting to tune into NASH, what do you want as the main takeaway, David?
I think everything starts at the level of primary care, or it should really. I mean, by the time they are seeing me for a liver biopsy, we want the primary care doctor to identify their metabolic risks, both to help their cardiovascular disease and as a beneficial side effect, address their liver. Also, that primary care doctor will make sure that their colon cancer screening is up to date. And in selected patients who are at risk for having Barrett's esophagus, which is a potential risk for esophageal cancer, to find those patients who might need a Barrett screening. So probably more education at the level of the primary care practitioner would be where I would start.
Great.
I wanna echo David, but with a little bit more tailoring to the liver. I want to ask the primary cares that it is time to manage this disease and decrease this epidemic and help our patients. I want to point the people around the world to the 2 documents by Diehl, how to risk stratify those at risk, such as type 2 diabetes or people with metabolic syndrome, or the AGA guidance. Just simple 2 pictures. Look at them, put them in your pocket, and start screening these patients, because not everyone should have biopsy. Indeed, now in clinical practice, when you refer the patients to us, we really do biopsy at minimum level. So the patients don't have to be scared. Teach your patients that this is a silent disease, but it can get people in trouble. And finally, if you end up sending the patients, you're helping them, and we have tools to help them, such as clinical trials, medications that they help the comorbidities, and the future is bright.
And Quentin, I'll give you the added opportunity to talk to policymakers and folks beyond the medical community about what they should be doing and what their takeaway should be.
Okay, I think that the key thing here is failure to recognize this disease is putting people at risk. So what I would like to see is established care pathways in each country, in each territory, triaging patients for having clinically significant liver disease. Now, that's not something that I, as an academic, am going to be leading on. This is a public health challenge. This is for the patient advocates. This is for the payers. This is for the people who make the policy. We need those pathways in place, because failure to diagnose is failing our patients. And we need to look forward to finding these people so that they can be treated effectively, whether that's with a drug or lifestyle intervention or just mitigating cardiovascular risk. Every one of those is a win.
Well said. Well said by all. Thank you so much for this conversation today.
Thank you, Donna.
Thank you for having us. Real honor and pleasure.
Thank you, Donna.
Please follow us, share, engage, retweet all of our International NASH Day panels. There are so many ways to engage. Find us on Global Liver on Facebook and Twitter, Global Liver Institute on Instagram, and use the hashtag NASH Day to tell us more about how you're engaging and spreading the word. Thank you.